Hello students, today I am going to lecture on the cells of the immune system. Introduction. A number of cells present in the blood are involved in the immune response. The white blood cells or WBCs are the major cells of the immune system. Among WBCs, lymphocytes are the central cells of the immune system. All cells of the blood arises from multipotent hematopoietic stem cells of the bone marrow. Hematopoietic stem cells differentiate into either a lymphoid or a myeloid progenitor cells. Lymphoid progenitor cells give rise to lymphoid cells and some dendritic cells. Myeloid progenitor cells differentiate into neutrophils, basophils, eosinophils, monocytes, mast cells, platelets, and megakaryocytes. Lymphocytes or lymphoid cells. Lymphocytes constitute 20 to 40% of the body's WBCs and 99% of the cells in the lymph. Lymphocytes are subdivided into three populations: B cells, T cells, and NK cells. Different lineages or maturational stages of lymphocytes expresses different membrane molecules called cluster of differentiation or CD. B lymphocytes or B cells. B lymphocytes mature in the bone marrow in a number of mammalian species including human beings and mice. Mature B cells display membrane-bound immunoglobulins, particularly IgM and IgD, as receptors for antigens. After antigen activation, B cells differentiate into effector antibody-secreting plasma cells and memory cells. Plasma cells secretes antibodies like IgG, IgA, IgM, IgD or IgE and have a very short lifespan of few days, but they can secrete more than 20,000 molecules of antibody per second. Memory B cells express the same membrane-bound antibody like naive B cells and remain in circulation for a long time and thus have long lifespan. B cells act as antigen-presenting cells. Mature B cells express many other molecules on cell membrane. Some of the important proteins present on the membrane of B cells besides antibody are CD45, MHC class II, CD21, CD32, CD80 and CD40. T lymphocytes or T cells. T lymphocytes arises in the bone marrow and mature in the thymus. Like B lymphocytes, T lymphocytes have membrane receptors called T cell receptors or TCRs. TCR recognize the process antigens, either antigenic peptides presented by mature histocompatibility complex or MHC on APCs. Like B cells, T cells expresses distinct membrane glycoproteins CD4 and CD8. CD4 expressing cells are T helper cells and those expressing CD8 are T cytotoxic cells. CD4 T helper cells are class II restricted and CD8 TC cells are class I restricted. The ratio of T helper and cytotoxic T cells is approximately 2:1 in the normal human peripheral blood. T helper cells are crucial for defense against pathogen and elicit different types of immune responses produced by cytokines. For example, subpopulation of T helper cells, that is Th1 response, activates TC cells and macrophages during inflammation, while Th2 response activates B cells during antibody-mediated immune response. Cytotoxic T lymphocytes or CTLs provide protection against intracellular pathogens such as viruses, bacteria, and parasites by secreting few cytokines. All T cells expresses CD3 associated with TCR and the following few other membrane molecules like CD28, CD45, and Di1. Natural killer cells or NK cells. NK cells are large granular lymphocytes that are distinct from TC cells. They constitute 5 to 10% of the circulating WBCs. NK cells are either B cells or T cells and lack antigen binding receptors. Therefore, they lack immunological specificity and memory. NK cells destroy the target cells by releasing the cytotoxic granules through ligand-receptor interactions. Unlike TC cells, NK cells kill the target cells in absence of specific antigen and destroy the malignant and virus-infected cells without prior exposure to the antigen. NK cells recognizes the target cells in two ways. Based on abnormal reduction of membrane MHC class I complex displayed and unusual markers on the cancer cells and virus-infected cells, and by process called antibody-dependent cell-mediated cytotoxicity or ADCC. Agranulocytes. Agranulocytes are non-granular mononuclear phagocytic leukocytes without lysosomal granules in the cytoplasm. It consists of monocytes and macrophages. Monocytes. Monocytes are the biggest type of WBCs and produced by hematopoiesis in the bone marrow. Monocytes circulate the bloodstream for about 8 hours. During this circulation, they enlarge, migrate into tissues and differentiate into microphages or dendritic cells. Monocytes are phagocytic cells. Macrophages. Macrophages are phagocytic WBCs that clean up the body by harmful and unwanted particles such as bacteria, viruses, and dead cells by phagocytosis. Macrophages are differentiated from blood monocytes. They disperse throughout the body in tissues. Macrophages, like cells, serve different functions in different tissues and are named according to their tissue location, as alveolar macrophages in lungs, histiocytes in connective tissue, Kupffer cells in liver, mesangial cells in kidney, microglial cells in brain, and osteoclasts in bone. Different types of cytokines like interleukin-1, interleukin-6, interferon gamma, and TNF-alpha secreted by activated Th cells and by component of bacterial cell wall enhance macrophages activity. Activated macrophages express high levels of class II MHC molecules to function as antigen-presenting cells. Granulocyte cells. Granulocytes are polymorphonuclear PMN leukocytes with 2 to 5 lobes. The granulocytes are classified as neutrophils, eosinophils, and basophils based on their cellular morphology and cytoplasmic staining characteristics. Mast cells are also included under the granulocytic cells. Neutrophils. Neutrophils have multi-lobed nucleus with granulated cytoplasm that stains with both acid as well as basic dyes. It constitutes 50 to 70% of the circulating WBCs and it produced by hematopoiesis in the bone marrow. They circulate in the blood for 7 to 10 hours before migrating into the tissues. Neutrophils are the first cell to arrive at the site of inflammation during immune response. Like macrophages, neutrophils are active phagocytic cells that release antimicrobial substances. Neutrophils also express Fc receptors. The granules in the neutrophils contain lytic enzyme and bactericidal substances. Eosinophils have bilobed nucleus with granulated cytoplasm that stains with the acid dye eosin red. Eosinophils are motile phagocytic cells that migrate from the blood to the tissue. Eosinophils constitute 1 to 3% of the circulating WBC. Their phagocytic role is important in the defense against protozoans and helminth parasites by releasing cationic peptides and reactive oxygen intermediates into the extracellular fluid. Basophils. Basophils has lobed nucleus and heavily granulated cytoplasm that stains with the basic dye methylene blue. It constitutes less than 1% of the total WBCs. Basophils are non-phagocytic cells. They are involved in allergic reactions. They have high affinity Fc receptors for IgE on their membrane. Mast cells. Mast cells are formed in the bone marrow by hematopoiesis as mast cells precursors or MCP. MCPs are released into the blood as undifferentiated cells and started differentiating after entering the tissues. Mast cells are found in tissue that are in close contact with extracellular environments, including skin, connective tissues, and mucosal epithelial tissue of the respiratory, genitourinary, and digestive tracts. Like basophils, they have large number of cytoplasmic granules that contain histamine and other pharmacologically active substances. Like protease, tryptase, and cymase, and preformed TNF-alpha, mast cells play an important role in allergic response by releasing histamine. They have Fc receptors for IgE and IgG. Dendritic cells. Dendritic cells are derived from myeloid and lymphoid lineage. Their main function of dendritic cells is the presentation of antigen to T helper cells. 4 types of dendritic cells are known: Langerhans cells, interstitial dendritic cells, myeloid and lymphoid dendritic cells. Despite of their differences, they all constitutively express high level of class II MHC molecule and members of co-stimulatory B7 family. Dendritic cells acquire antigens by phagocytosis, process it and present it to T helper cells. Now I would like to conclude my lecture by saying these few points. All leukocytes or WBCs differentiate from a common multipotent hematopoietic stem cells during hematopoiesis with the help of various cytokines. Lymphocytes are the major cells of the immune system and they have 3 types: B cells, T cells, and NK cells. NK cells are less abundant than B and T cells and lack receptors for particular antigens. However, NK cells have both TCRs and other markers characteristics of NK cells. All types of lymphocytes are best distinguished by their presence of various membrane molecules and function. Naive B and T cells proliferate and eventually differentiate into effector cells and memory cells after antigen activation. Macrophages and neutrophils are specialized phagocytic cells that degrade antigens. Macrophages process the phagocytized antigens and display it with MHC class II molecules to T helper cells. Basophils and mast cells are non-phagocytic cells that play important role in allergic reactions by releasing a variety of pharmacologically active substances. Dendritic cells express high level of class II MHC molecules, and dendritic cells engulf antigens, process and present the class II MHC molecules bound antigens to T helper cells. So these are the cells that we are studying today for the cells of the immune system.